Good. Welcome, everyone. Good morning, good evening, good afternoon to all of you joining from wherever you're joining. I hope you're staying safe in the midst of these very uncertain times. I'm Pat Furlong from PPMD, and I'm here with Linda Marban from Capricor Therapeutics. Today, this webinar is going to talk about CAP-1002. This has been a study that first started with HOPE-1, which will be lightly touched on in HOPE-2, and we'll concentrate on the results. Linda Marban is the CEO of Capricor Therapeutics. She's been with the company and is one of the co-founders of this company. She combines her basic research entities and interests to really create a path for commercialization of this novel therapy in Duchenne muscular dystrophy and other areas. So I just want to talk for a minute before I turn this over to Linda. This was a study on patients who were older than what we typically see in Duchenne studies. These are patients that were in transition about losing ambulation or have lost the ability to walk. The focus of the outcomes of the study was the PUL, the performance of the upper limb. The performance of the upper limb is really a measure that we are looking at arm strength, and it's divided into three different domains. The shoulder domain is important, and this would be for patients to be able to assess whether they can reach up and turn on a light, or whether they could raise their hand in class, whether they could hug their parents by reaching up. So those are some of the important issues in the shoulder domain. In the elbow domain, there are other important issues, and improving in any of these domains is really incredibly important to the non-ambulatory population. So as you might imagine, in the elbow domain, it is about reaching, right? It's about reaching for things, being able to really take care of yourself if you're able to have good elbow flexion. And that, in other words, is really important to people who are non-ambulatory to move in their space and have good arm extension. The wrist is the third domain, and this again we would all think about that. That's for computers and for technology and for phones and to be able to manipulate a glass with your hand. So all of these in the PUL, and this PUL outcome measure was designed by patients and families with non-ambulatory patients who had limited or were decreasing in arm strength. And so we really wanted to assess what's of value to them in the PUL. So improvements in any of those domains then make sense and improve the quality of these patients' lives. The other areas that we explored in this study were the heart. The heart is a muscle too, and we must all remember that, and we continue to want to preserve and protect the heart function and certainly improve it or prevent further decline. The heart function, the heart at this current moment does not have any regulatory acceptable outcomes, but certainly we know strain is present on cardiac MRI from a very early age, often as soon as we can get these little kids in the magnet, and continues and has a real relationship to when the ejection fraction declines, as well as the pulmonary status. So these are the global sort of interests of this study. This is a hard population to study because it is a more fragile population and often one that declines very rapidly. So this study really focused on that and asked the question, can we change that? Can we delay the progression of this disease, and how will we measure it? So with that, I'm going to turn this over to Linda Marban to start talking about what Capricor has done, what we've done in Duchenne, and what we've seen. So with that, Linda, please take over.
Thank you so much, Pat. And I want to say thank you to Pat, to Parent Project for Muscular Dystrophy, for all of the families, all of the patients that have made themselves available for this study. I have such admiration for all of you who fight every day for your children, for yourselves, and to make the world a better place. So let's hope that we all can do that together. Okay, so we are a publicly traded company. We're listed under the symbol CAPR, and this is a standard forward-looking statement slide, which basically says that our filings are up to date and can be accessed through the Securities and Exchange Commission. So Capricor is a rapidly growing small biotechnology company that started on the foundational observation made that CAP-1002, or cardiosphere-derived cells, have impacts on cardiovascular function. They are not, which I'll talk about in a few minutes, but they mediate their impact by stimulating tissues to repair themselves and to suppressing inflammation. From that program, we have now spread out into areas of clinical investigation in COVID-19, When you think about the inflammation that occurs in Duchenne, it's not dissimilar to the type of inflammation that occurs as the post-viral sequelae of the coronavirus. So we're working on that as well. And then we've isolated out the mechanism of action of our cells called the exosomes. I'll talk a little bit about those today as well. And they're a unique product. We are actually looking to expand into the younger children with the exosomes with Duchenne over the next 6 months to a year. And then the rest of the exosome program is on platform development of engineering the exosomes, and I won't talk about that today. But if you're ever interested, you know where to find me for more questions. So let's talk about the technology briefly. CAP-1002 is actually the developed product of cardiosphere-derived cells. They are made from heart muscle, human heart muscle. We get tissue from hearts that could be transplanted but are not, usually for technical reasons. We isolate out our cells of interest from there and grow them up in proprietary means. These are not stem cells. They do not go into the injured tissues and repair them directly. What they do is they stimulate the release of exosomes. Exosomes are nanometer-sized lipid bilayer vesicles, which is the scientific way of saying that they're extremely tiny little pieces made by cells coated with cell membranes that are used for communication by cells. And we've been able to identify the exosomes and their contents that drive the potency of this product. CAP-1002 has been in over 150 human beings over the past 2 years, so we know a lot about it. Next slide. So I'm just going to spend a few moments on this busy slide, which summarizes the preclinical work that we did in preparation for treating patients with Duchenne muscular dystrophy. As all of you are well aware, the animal model where all of these studies usually begin are in the MDX mouse. It's a naturally occurring mouse model of similar disease to muscular dystrophy that's sort of good enough for making preclinical observations. And what we're able to do then is to study them to see if what we're trying to develop makes any difference. What you see here in this slide are basically 4 ways in which we studied the mice. We were able to show that we had an impact using our cells, CAP-1002, to improve cardiac function or ejection fraction, and you're now going to see ejection fraction data in people, which shows improvement. We were then able to show that the animals could run on a treadmill longer. This is the mouse analogy to the 6-minute walk test. The bar up here are healthy mice, and then the 2 bars down here show the treated mice compared to the placebo mice, and you can see that they're able to run farther on a treadmill. We then took out muscles from their legs. It's called the soleus muscle. It's kind of similar to our quadricep muscle in our legs, and we were able to put them in a muscle bath and show that the treated animals had much better muscle function independent of anything else compared to the placebo-treated animals. And finally, we looked at the diaphragm, and it's almost impossible to measure the function of the diaphragm in terms of its ability to move or contract, but we were able to look at fibrosis or scar in the diaphragm, and we were able to see that those were also improved in the mice. So I want you to put this away in your memory, and because when I show you the clinical data, you're going to see this is one of the first times that the preclinical data predicts the clinical data. So cardiac function improvement, overall skeletal muscle improvement, isolated skeletal muscle improvement, and lack or reduction in scar in the diaphragm. So the first clinical trial we did was called HOPE-Duchenne, and HOPE-Duchenne was in patients that had cardiac scarring as their inclusion criteria. And these patients came into the study, and because they were farther along in the disease process, they were primarily non-ambulant. So we had to look for a way to measure whether their skeletal muscle function was impacted. And as you know, and Pat just talked about, once DMD kids come off their feet, there was really no way to assess that into the development of this measure called the performance of the upper limb, which I'll talk about in a few minutes, which measures the ability of the shoulders and the arms and the hands independent of the legs. And what you see from the HOPE-Duchenne data, which is very interesting, is when we look at MRI, and this shown here is the donut type shape, which is the heart. White is bad, it's scar. Black is good, it's healthy tissue. This is a CAP-1002 treated patient at baseline, and when you look out by 12 months, you can see there's a lot of holes in his scar. His scar is actually reduced, which should lead to improved functional implications of these patients. These patients got one dose of 75 million cells, and this was a study done intracoronary, which means these patients came into the cath lab, and they had a little tube pushed up into their heart where the cells were delivered directly. This is an MRI of a usual care patient. They didn't undergo this procedure. They start off with a lot of scar. The scar is pretty much the same at 6 months. And while this image is a little bit cloudier because sometimes the children move, you can see there's a lot of scar still at 12 months in this very same patient. We then pooled the data and we looked at it and we saw that on average those patients that were usual care, they were randomized and not treated, had the same or more scar in their heart at the end of the year, whereas the treated patients had less scar at 6 months and a year later. This was reflected by looking, as Pat said, some of the MRI technologies allows us to look directly into the heart, and we were able to see inferior wall thickening or improvement in the area where the scar was getting thinner. And then the most important defining piece of data was the improvement in the performance of the upper limb in the patients. What you can see is, remember, one dose of cells. We studied them for a year, and you can see that the patients that got the one dose of cells did better, and they really did much better in the first 3 months. So this defined our clinical program going forward, which is that we would be not having to use intracoronary delivery anymore, and I'll talk about that when I get to HOPE-2. So, DMD is an orphan disease. Everybody knows that on this call. So we have orphan drug designation. That's very important. It allows for sort of committed resources to be allowed to be developed for use in orphan disease. We qualify for a rare pediatric disease coupon. That's good because if we get approved, small companies can sell that and use that to fuel their engines to develop the product for commercialization. And we have RMAT, or Regenerative Medicine Advanced Therapy designation. This is breakthrough designation for a cell or gene therapy. Very important because it allows you preferred access to the FDA. They really work with you on development of protocols and plans so that you can get to market with a cell or gene therapy faster. I will talk to you now about the HOPE-2 clinical trial and why we think this data is so transformative for the Duchenne space. Let me just remind you very quickly, the patients that we are focusing on. We are focusing on those non-ambulant patients or early-stage non-ambulant patients. That means they're about to go off their feet. These are 50 to 60% of you out there with Duchenne, and yet the most untargeted section of the population in terms of therapeutic development. Even to this day, with as much work as PPMD and some of the other organizations have done to call attention to the fact that just because you're off your feet doesn't mean that you have any less interest in living life and maintaining independence and privacy. Most clinical work has been done on the younger children to see if we can delay the progression of the disease and keep them walking. We take a different approach. We say, you are off your feet, but you still have a whole lot of living to do, and let's see if we can keep you stable or make you better so that you can continue to live life and stay very active in your own world. So let's talk about HOPE-2. HOPE-2 was a randomized, double-blind, placebo-controlled trial. In the participants with Duchenne, you had to have been diagnosed with Duchenne specifically and a reduced skeletal muscle function, suggesting that you were not going to be ambulating for much longer. It was a safety and efficacy trial. This trial, we were using 150 million cells. Why did we double the dose? Because we showed that we could if we went to intravenous delivery as opposed to intracoronary delivery. So the patients in HOPE-2 just had to sit up and get an infusion intravenously as opposed to go to the cath lab. We were at 9 Duchenne certified sites across the United States. And the trial was only done in 20 patients. Originally, we were planning on doing 80 patients. We had to cap early because of financial constraints. What we're very lucky in is that the data already is telling such a strong story that we're hoping to convince FDA that it requires approval so that we can make this readily available to all patients with later stage Duchenne. The patients were 14 years of age and old on average, and some younger, some older. All were on stable doses of corticosteroids, and 80% of you were off your feet. So the performance of the upper limb, as Pat talked about, and I'll give some details on in a moment, is a way of measuring by patient-developed, physician-developed, physical therapist-developed ways of looking at activities of daily living, things that make your own independence and privacy so valuable, but in a quantifiable way. So we can actually measure it and qualify it and validate it and use it as an endpoint in a clinical trial to say, yes, you're getting better, or no, you're not getting better, or potentially getting worse. In this particular trial, we used those that had an entry pool score of between 2 and 5, so not so low on this entry item score that you may not be able to get back some of the use that you've lost, and not so advanced that we would not be able to see improvements at this point. So sort of in that Goldilocks zone, a sweet spot. The performance of the upper limb, the version 1.2, which is sort of shown here in terms of this fleshed out strategy map or skill map, is the things that you are asked to do that, as I mentioned, are analogous to the activities of daily living. They start with shoulder and they end with hand and wrist. The 1.2 version was the first version that was developed. It was developed basically, again, by the hue and cry of the community that once patients were off their feet, didn't mean that they were off their lives. And so we were able to take advantage of that and develop this very important assay. The 2.0 is the better version. As I always tell people, it's like your smartphone. You always want the newer, improved version. The 2.0 is therefore somewhat better than the 1.2 by having better strategy compensation allowed. The response scale is different, which actually reduces variability. And the floor and ceiling have been attenuated, so we're really able to look at change, and it's a stronger, better assay. So let's talk about the data. First, we're going to talk about the upper limb data, the performance of the upper limb. I will tell you, and if any of you have had a chance to see this data, it is quite exciting. This is showing the full pull, the performance of the upper limb, version 2.0, the shoulder and the mid and the distal combined. This new version of the pull really takes advantage of all three muscle groups so that the full score is actually truly reflective of where each patient is. Shown in blue are the patients that were treated with the cells, and shown in red are the patients that received placebo. And what we're showing is the mean change from baseline, so each patient has his own baseline measured, and then we looked at him over the course of the year, and this is at the end of the year. You can see that the placebo patients had a decline that has been published and associated with typical decline in a patient with Duchenne over the course of a year in upper limb function, whereas that decline has been attenuated by more than 50% in the patients that were treated with cells, suggesting a decline that would be not even considered statistically relevant when compared with a normal non-change, or based on standard deviations, shown in this scatter plot. And I will tell you, I think many of you know, I'm a PhD scientist by training, and when I look at scatter plot data, it usually makes me cringe because we don't see it as reflected in the mean. And what we can see here is that, in fact, this is a scatter plot that is truly reflective of the therapeutic efficacy of this product. So it's well worth showing and shows the importance of the improvement to each individual patient. Again, I don't get to show this data very much, so it excites me to share with you the individual patient data. This is the trajectory patients. And this is why we're hoping we can get this approved, because the patients really go through a lot in these clinical trials, and my respect goes out to the children or young adults that participate in this, as well as their families, because they have to travel to centers every few months and be measured. And what you can see in the treated patients, the decline was much attenuated compared to the placebo patients. All of the placebo patients are going down steadily over time with some changes here and there. Sometimes the child will have a bone break or something that changes their performance at one time point or another. But in general, you see improvement. This patient here, the patient that did the worst in the CAP 10 or 2 group and has actually driven the averages down across the board, was a patient that has a pretty severe manifestation of Duchenne and went off his feet very, very young. So we're hoping for his continued well-being, but was considered pretty much a non-responder in this study. And then finally, for those of you that have been following us for a while, the original trial was powered against the mid-level pull 1.2. That's the elbow. In the first version of the pull, some of these subsets of measurements were actually potentially more valuable in looking at improvement in function. We didn't hit statistical significance here, but it's pretty darn close, and we actually saw a 2.8-point difference in the CAP-1002 treated group versus the placebos at a year, which suggests that we have a real clinically relevant treatment effect. And look for more information on that. That data is just rolling in now, and I'm very, very excited by what it shows. Shown here again in the scatter plot is the patient that I showed you in the previous spaghetti plot, who really is an outlier in terms of his functional ability. And again, this is the spaghetti plot, individual patients that were able to be measured. Again, this is our non-responder patient. This young man had a very nice improvement and may have had a drop-off after a year if he had had something happen to him, but was really doing well. What's interesting, and I think even more relevant, is that the placebo patients follow the standard decline. And remember, these are real placebo patients. This is a double-blind trial, so nobody knows what they're getting. What we think is very interesting is that in the placebo group, there were more dropouts than in the CAP-1002 group, suggesting that families might not have been seeing a difference and kind of got discouraged. Don't get discouraged if you're in clinical trials. Your participation is always important no matter what group you're in. We need it. The regulatory agencies really care. So let's talk about cardiac function. I got to know Pat now several years ago. I'm grateful that we get to grow older together, not old, but older. We came to her originally because we thought we had something interesting in terms of a cardiac function therapeutic. It was just when she and her colleagues on the medical side were coming out with their white paper that talked about the impact of cardiac function on Duchenne patients, and that a significant portion of patients when they're dying from Duchenne are now dying from implications of cardiac dysfunction. And what I can say is that we see real opportunity here for preservation of long-term cardiac function. This is ejection fraction. Ejection fraction is considered in the space the gold standard for improvement or for measurement of cardiac function. It's how we would quantify or classify an adult with heart failure or a person with a cardiomyopathy. It is sort of the indicator, the gold standard indicator of cardiac function. For those of you that don't know, I'm a cardiac physiologist by training, so for me, cardiac data is something that is near and dear to my heart, and this is the type of data we almost never see with p-values of 0.004 in terms of ejection fraction improvement or preservation of function. Most of the patients in HOPE-Duchenne and in HOPE-2, the patients that we have here, had cardiac function that was not in the dangerous zone, but we'd like to keep you from getting into the dangerous zone. And if we continue on this trajectory, and what we'll hope to show in a long-term confirmatory trial is that we can keep your heart function stable for a long time and significantly reduce the opportunity for mortality based on cardiac dysfunction. This is the individual patient data again. Again, I like to show it. These patients, you're only in the magnets a couple times at baseline, 6 months and 12 months. And you can see that the, above the zero line patients that are staying healthier or even getting somewhat better in the CAP-1002 group is irrefutable, and yet most of the patients in the placebo group are on a steady decline, which is typically what you'd see in cardiac function after a year. As Pat said, it actually starts when the patients are very young. It just is not readily visible. And remember, many non-ambulant patients don't really feel their cardiac function because they are not putting a huge demand on their heart. So left ventricular end-systolic and diastolic volume, these are index values. That means we take body surface area into account. This is important because, as many of you know, from chronic steroid use, there's some changes that occur to body shape. It's called dysmorphia. And that basically allows us to really look exactly at cardiac function based on your body as opposed to on a standard. In this situation, down is better. So let me just tell you for 2 minutes about cardiac function. Cardiac function is a really beautiful thing. The heart is based on really very simple principles of physics, which are pressure and flow based. and you have to generate enough pressure to eject enough blood out of your heart to be able to feed your toes and your brain at the same time. If the dimensions of the heart change, just like if you think about your garden hose, if you put a big, if you fatten out your garden hose, you're going to get less flow. Such is the same with the heart. So as you change the structure of the heart, so you change the function of the heart. And what the volumes tell us, end-systolic volume is the amount of blood that's left in the heart after you've had one beat or one contraction with the blood ejected from the heart, and it's a good measure of size. And diastolic volume is basically the filled heart. Right before you can measure this, you can capture it, the blood is ejected. And these are all very important measures for determining development of the appropriate pressure so that flow can be sustained. What you want to see here is the heart going more back towards a normal size. And in our treated patients, you see that we actually saw volume decrease. Where in the placebo patients, unfortunately, it's increasing. Think again about your garden hose. If you start to expand it, your flow goes down. We want to keep the size of the heart the same or potentially make it better. And we've seen in both indications here, the end-diastolic volume and end-systolic volume, that we're making it better. We even have statistical significance in end-systolic volume. End-systolic volume is one of the few surrogates that's ever been used as an approval endpoint for adult heart failure because it's such a relevant measure of cardiac function. So let me just say, as a cardiac physiologist, I look at this data and it almost makes me cry because it shows real opportunity for preservation of cardiac function in a patient group, and isn't that why I went into science to begin with? And then finally, sort of what I would consider the frosting on this beautiful cardiac cake is that we actually have a biomarker that is reflective of the preservation of function. So let's talk about this for a minute. Creatine kinase is an enzyme that's made by muscle. You have basically three types in your body: MM, which is skeletal muscle; BB, which is for the brain; and MB, which is released or created in heart muscle. Typically, in normal, healthy people, you don't see much creatine kinase in the blood. If you're a runner or do heavy exercise, you might see some MM from breakdown of skeletal muscle. As you can imagine, Duchenne patients have a ton of creatine kinase in their blood. Their MM is typically high. Their skeletal muscles are breaking down. And you also see more MB because you have cardiac muscle breakdown. Remember, this is a dynamic process where cells are being broken down. But unlike skeletal muscle, you really don't get a lot of replacement of cardiac cells. So you don't get second, third, and fourth chances. What we see here in this creatine kinase MB fraction is we see that we have significant amount of MB circulating in the blood of our placebo patients, where we actually see almost no MB or a significant reduction of MB to a 0.006 level in the blood of the treated patients, supporting the concept that cardiac function is being improved and there's less scar, as we saw in the original HOPE study. And this is, again, the individual patient data. And again, you can see, that the placebo patients are on average going up, almost all of them, and on the CAP-10 tubes, they're going down. So we also looked at respiratory function. We mostly were focusing on respiratory function as it is measured by muscle function. As all of you know, measurement of respiratory function in Duchenne is a real bugaboo because forced vital capacity, the gold standard, is hard. You have to get height. Non-ambulant kids, you can't get height. Then you have to use ulnar measurements, which is a bone in the arms, and that's very hard to do. And if you see even small changes in the ulnar measurement, it can change forced vital capacity. So it's a moving target that's not very easy to see. We looked at two measures specifically: inspiratory flow reserve, or what's left after inspiration, and this is a measure of inspiratory muscle function. And you can see again in these individual patient data that the CAP-10 patients are largely doing better. The placebo patients are pretty similar, superimposable, but definitely getting worse. And over time, this is exactly what you would expect to see in a placebo or a non-treated patient with Duchenne, that their muscle function in their lung and their chest wall is getting worse. And then peak expiratory flow percent predicted. This endpoint was called out by the Food and Drug Administration as one they would have liked to see in our data set when we went in before we started the clinical trial. This is really, again, a measure of muscles that are used in expiration. So, hopefully quietly, or if you're on mute, you can breathe in hard, inspiratory flow reserve, breathe out hard, peak expiratory flow, percent predicted takes, again, height into consideration, because we have to make sure that we're not comparing patients to normal developing children, because ours are steroid treated, as you know. And so that's sort of the efficacy data taken together, a very exciting data package. I'll talk about safety for a few minutes. Of course, we and of course the parents are most concerned about the safety of their families. There was a lot of hoopla from us as well as the community when everybody heard that we went on voluntary holds in early 2019 after we had an anaphylactoid type response in late December of 2018. What we learned is that this is not uncommon when biologics are infused into patients. What we did is we did tremendous amount of data diving with the patient that was impacted, and then there was also one patient in the original HOPE-OLE trial. And we were not able to find real cause. It may be linked to excipients, we're not sure, but what I can tell you is that we've been able to remediate this by starting a pre-medication regimen, which in Duchenne patients are steroids and then antihistamines orally given, and not so very impactful to patients and families, and thankfully we haven't had anything since then.
So, Linda, I'm going to just stop you right there just a minute before I know we're going to let you complete, but so go back to the previous slide and just talk about excipients. I think that people on the line may not understand what that meant, excipients,
yes.
and what that meant, and how those excipients this is not unusual in a stem cell application outside of Duchenne. So I think if you'll just explain that a bit, that'd be great.
Yeah, so basically, there are always excipients in cell therapies because we have to make sure that the cells don't clump. And so we have to have things in there like sugar, like DMSO, which prevents clumping, sometimes albumin, which is like a protein, which also helps the cells stay dispersed and go into the body appropriately. So there are always excipients in these things, just like any drug. Even if you take an ibuprofen for a headache, there's things in there that will be the active ingredient and then the non-active ingredient that make you able to swallow it or whatever. So they're in these drug products, and the patient that experienced it was a cell-treated patient. But when we did allergy testing on him, we didn't see that he mounted in response to the cells. And because it happened so quickly, within 1 or 2 minutes after the infusion began, we know that it's unlikely to be an immune response. The good news is as we just talked about, patients will always get this procedure in a medical setting. And so, they'll be observed, and nothing has ever happened after the first few minutes. So in the one patient in HOPE and HOPE-2 and the one patient in open label extension, they had the response within a few moments. And so, we'll always want them to be in an infusion center, but we haven't seen anything since. And both patients, by the way, recovered immediately, went home the next day with no sequelae. So this was something that was definitely something we needed to address, but not something that should prevent this product from moving forward.
Great. Thank you.
Yes, of course. Thank you, Pat, for that clarifying question. Since that time, we've done 42 infusions, and we had one patient that we had in the hospital overnight. We're not convinced that it was the same type of hypersensitivity reaction because it was very different in its manifestation. He was fine and went home. So where does that leave us? That leaves us really excited with this data. This is a segment of the population, as I said at the beginning, of Duchenne patients that really may not qualify for a lot of the gene therapies. Dr. Lee Sweeney, who is somebody that I've gotten to know through Pat, and also has become somebody that I respect tremendously in his ability to understand the global implications of therapeutics, has talked a lot about the fact that perhaps patients with advanced Duchenne have such fibrosis in their muscles that it may not be possible for the gene therapies to make a difference. Regardless, it's going to be a long time before those are available. This is a here and now therapy, and if we can delay the progression of the disease, and keep patients from losing their opportunities for privacy and also for independence. This is our goal as a company right now. What we're doing is we're going to work with the FDA. We really hope they won't ask us to do another clinical trial, especially in this day of COVID-19. I'd be interested to hear families' impressions, but I know that if I had a child with a pre-existing condition, I would not want to take him or her on an airplane every quarter for a therapy and a clinical trial, so it could markedly delay the opportunity for getting this commercialized. We are going ahead with the open label extension, making that available for all patients in the trial, whether they were treated originally or placebo. We feel that we owe it to the patients and to the families that have put themselves out there, since we are seeing such good impact. And so we look forward to continuing to update you on our progress. And please, let's all work together to get this approved as quickly as we can.
So Linda, back to that one again.
Yes,
I have a few questions, just some clarifying questions. So you have these patients on open label extension, which is pretty terrific, and I'm sure they're grateful for that. So if you could close your eyes, give us a timeline. What are you hoping for, recognizing that all of this depends on whether the FDA is willing to accept the application and so on. But how do you see this going forward? You're obviously going for an accelerated approval. When and what is the time frame for that approval? That's my first question. The second question is, in terms of production of these cells, you're giving 150 million cells every 3 months, as you've described. So it's not like a loading dose and then it changes. So it's 150 million every single 3-month period. Do you have enough capability to manufacture? We don't have actual exact statistics, but I'm guessing that there's 60% of the population of Duchenne is off its feet in general or in transition, or greater numbers than that. So, we're looking at thousands of patients. Can you manufacture enough? Can you deliver enough? So first, your timeline, if in your head, if things line up, and second of all, how are you going to manage to deliver?
Yeah, so thank you, Pat, for both of those. I'm going to take them in reverse order. In terms of the manufacturing, we are now working with Lonza, which is a worldwide contract manufacturing organization, to make the doses for commercialization. Once I was convinced that once we did the interim analysis and I saw how powerful that data was, we entered into that tech transfer agreement, and that's going along beautifully now. So yes, we will have enough doses to provide 150 million cells 4 times a year for as many Duchenne patients as want it. And of course, we hope to go international with this as soon as we can get a trial going in the EMA. All of this is based on resource management and capitalization, not lack of desire. We are unfortunately small and have to go only as far as the dollars will take us. But that's something that we are working on actively now, is the tech transfer for commercial doses, and we anticipate being able to meet the need, in a very real way. That was one of my goals as CEO. In terms of timeline, it's all dependent on the FDA. I'll give you my dream version. My dream version is that we meet with the FDA very soon. That's imminent. That they tell us that contingent upon the tech transfer plan, the CMC, which is the manufacturing aspect of the development of the cells, and then for commercial use, and then also probably asking for open label extension data. Would I be the FDA? That's what I would ask for. I would anticipate preparing a BLA by the beginning of 2021, and then commercialization as soon after that as we can get approval. Now that's the Linda Marban version. It's possible that the Food and Drug Administration may stamp their feet and say you need another clinical trial. Why might they do that? Because of the interim analysis that we did, which broke our statistical analysis plan, the FDA considers this an unblinded study, and so they don't consider us having hit our primary efficacy endpoint. And in traditional clinical trial design, that's not how one would want to move forward. They also may ask for more safety data. That we've tried to hedge by doing these very sick patients with ARDS and COVID-19, showing the safety of the product in even very sick, respiratorily compromised patients. But that could slow us down depending upon what FDA says. If FDA is willing to let us move forward in the timelines that I have suggested, we could potentially be providing doses to patients, in a very short timeline, and hopefully those patients that we're treating today will continue to improve or stay stable. If we have to do another clinical trial, I'm guessing it's a 4-year delay to getting this to patients in a more global fashion. And I don't mean global as an international, I mean as in any patient that's not in a trial.
Right. So, Linda, sometimes, as the trials, as a particular study ends, what happens is that even if it's in an open label extension, the data collection is not as rigorous as it was during the study. So in your open label extension, are you collecting the data in a rigorous way so that we can see that these patients remain stable or improve? So that's one question that I have. And the second thing is I'd like you to explain a BLA to the group on the phone, what that BLA is. And also the process. So you're going to, and also a couple things. One is you went to the FDA with interim results, and that changes what the FDA's perspective is on the study itself. That basically says the FDA is saying now you've read the results, and so there's no more blinding in the study. So it's not the rigorous study that we're requiring, and then we're going to have to do another study. So if I have that correctly. So talk to us about going back to the FDA, what the BLA is, and should they then say, okay, you can submit the package, then their timeline for approval, and just go through that a little bit more methodically, if you don't mind.
Absolutely, thank you. So first, the BLA is a biologics license application. It's like an NDA, a new drug application, which you may be familiar with from some of your opportunities with Sarepta and other companies that have gotten approval. What it basically is it's your full dossier of everything you've ever done with this therapeutic that leads them to decide whether they can approve it for commercial use. So it's all your safety, it's all your efficacy, it's all of your preclinical work, it's all of your toxicology work, it's how you manufacture your cells, how you make sure that batch-to-batch consistency is maintained, your potency, everything you can think of. And it's a huge document that takes many months to put together that the FDA then reviews very, very carefully and bases the determination for approval on. If you submit a BLA or an NDA and they can't really decide, you can request an AdCom, which, as you may remember, Sarepta had and BioMarin had, where experts are gathered to determine whether or not the data supports registration and therefore commercialization. So stay tuned on that. The timeline for that is about a year from deciding to submit to submitting and getting all the meetings in, although that depends, everything's changed a little bit now because of this pandemic. In terms of the open label extension, Pat, you are exactly correct that many times this is not done in a strategic or surgical way, but we have decided to do it in a strategic and surgical way. We knew that because we had stopped this trial early that every bit of data that we get would be important. And so even though our trial is considered unblinded because I have seen the data and I've seen individual patient data and know who those patients are, the patients don't know who they are. So when the families that are on today look at the lines on the spaghetti plot, they don't know which group they're in, and they don't know if they were treated or untreated. A lot of families have suspicions, but they don't know. And so what we're going to do, and that's why we decided to treat all patients in the open label extension, is that we don't have to unblind anybody. So they're going to still be blinded to their treatment assignment. And they've been off therapy long enough that we're hoping to see this as sort of a crossover design. We're going to be doing video collection of activity of the patients as well as the performance of the upper limb measurements so that we can quantify their improvement in a very clinical trial sort of best possible opportunity way. And so, yes, theoretically, patients will be more excited to get this, but none of this can be attributed to volition because they don't know whether they have actually received placebo in the past or cells. And so this will be something that we can actually measure in a quantifiable way. Did that answer your questions, Pat? Did I miss one?
Yeah. No, that's great. There's another question. Of course, I have thousands of them for you, Linda. So, Linda, Irreversible, which is a company that is conducting a trial, has an NIH grant to deliver patient-specific results back to the families who participated. Do you have any plans, and I know you can't do it at this minute, but would you have any plans or would you be willing as a company to return individual results to a family, what particular arm and what the results were along the way. I think this
Absolutely.
is really coming from the patient community about how did my son do, right? That's obviously so important to every family.
Absolutely, and I can't emphasize enough that we want to work with the families and make sure that they know. It's important to stay blinded now, and that's why we decided to dose everybody again. But what I can tell you is once we get to the end, especially since we have a cardiac component, I get calls from families frequently saying, oh, my son had an MRI with you, what was his ejection fraction? Families are worried with their later stage patients about their cardiac function. So yes, we will make it available. I absolutely promise that. I know there have been patients from the original HOPE-Duchenne trial that did not get their results. I think that was largely because the PI left the medical institution where he was, and somehow it has to be delivered by the physicians, right? I can't do it myself, but we will make it available to the physicians so that they can make that available to the patients, and we are very committed to each individual family.
Yeah, it would be really important for all these families that were in the trial to get a note from the PI, just that we're going to do this. Timeline hasn't been established because obviously you've got a regulatory framework that you need to fulfill. But I think just reaching out to them with information that is coming and they know about, that would be great. I think a couple things. One question we have is, do you have an FDA meeting scheduled to discuss this? Is that you can just say yes or no and not give us the date?
Yes,
if you have
we do.
Schedule.
Yes, we do.
And there's some questions on sort of what these exosomes are doing. They're anti-inflammatory, they're antifibrotic, but other than within the imaging DMD study, we don't really have any data on the level of fibrosis in these individuals who participated in the study. I mean, we have it in imaging, but we don't have it on these individuals, and you did no biopsies. Is that correct?
Yeah, so we decided not to do biopsies. They're painful for the patients, and they don't necessarily reveal a lot. We've seen in many other clinical trials sort of the fall down of the therapy because it probably works, but we couldn't quantify it, and so we wanted to stay away from that. But we do have it in the animals. We have a lot of preclinical data to suggest that fibrosis is reduced. We've been able to show stimulation of generation of new tissues. I've been able to demonstrate that exosomes are taken up by the satellite cells. We see reentrance into the cell cycle by Aurora B measurement, immunohistochemistry, which is a way of taking pictures of things that happen in a sub-visual or microscopic way, and other ways of measuring fibrosis in the animals, but not directly in the children.
Okay, thank you. And if you'll go back to slide 23 for me, that would be helpful.
Okay.
Yeah, it's about the CKMB.
Yeah, I'm there.
So I think there's not a person in the Duchenne environment that doesn't know about CK, right? It's often the diagnostic measurement that alerts the primary care that there is muscle wasting going on because we see the CK muscle going up to the thousands, sometimes 20. And I remember when my sons were diagnosed after an injury, when the day Chris was diagnosed, his CK was 70,000. So the CKMB, I don't think we as a community have really been educated ourselves on the variations of the CK. Clearly, I'm pretty familiar with them having been here, but do you have data in the Duchenne community? Is this something, a biomarker that others have commented on? Is this something, just in your opinion, as our community, we might, as a community, go to the FDA and talk about CKMB as a potential biomarker? As you know, we've had PPMD supported many meetings with various cardiologists and certainly with FDA to talk about we need a really acceptable measure in the heart so that we can understand decline, but moreover so that we can get drugs that preserve this heart approved in a rapid way. So did you have any fundamental data on the CKMB and what would be your recommendations to us?
So there's very little, of course, when I saw this data, this was one of those situations, and I'm always very honest, it blew me away. I almost started to cry when I saw it, and the chairman of our board is a cardiologist, and he had the same response. Those of us that work in adult cardiology know that this has always been considered a gold standard of cardiac muscle breakdown, and it's nice because it's very specific to the heart. Now, we've moved on as a world in the field into the troponins, and we're gathering that data on our Duchenne patients as well. But as you know, Pat, that measurement is extremely labile. So troponins may be impossible to predict clinical progression because, as you know, some of the kids have rises in troponin that don't seem to be impactful or impacted by anything. So this was a really nice opportunity. There's almost no published data on CKMB and Duchenne. There's a lot of published data on CKMB and adult heart disease, and that data goes back 30, 40, 50 years because it was one of the very first things we could use. The CKMM, which is the skeletal muscle fraction, we also have that data. We've seen changes in the treated patients compared to the placebo patients, but I've been less apt to present that data because if you have an active day as a human being, you may have more MM in your blood, and so it's one of those situations where I just don't think it's going to be as quantifiable. And the BB, what I can tell you, the brain version in all of the patients measured had 0. So from baseline all the way through, we had none. So the answer is not a lot of data on this in Duchenne, something that I find fabulous and interesting from an adult cardiology perspective, but this is something we're still trying to learn about.
Yeah, we should go back and take a look and see what's been collected over time. I think that so there's a question about CK and just as a sort of placeholder here, the CK is what we measure. The CKMM is what we really measure most of the time, and that is asking how much muscle, skeletal muscle deterioration is going on. And that varies with activity. It varies with injury. Like my son went up to 70,000 when he was diagnosed because he had he had an injury in his calf muscle. So the CK just spiked that next day to 70,000. But in general, I mean, CKs are all over the map. We tried at one time to say, is CK a good biomarker? And it isn't because it depends on the day, the time of the day, the age of this patient, lots of factors that can affect it. The CKMB is really very specific to the heart. So we have not ordinarily or has generally been collecting this in Duchenne, although my guess is if we go back and look in some of these clinics, they might have collected that in some of what they've been looking for during the cardiac MRIs or whatever. So we might want to go back and take a look and see what's there. So Linda, I know we're getting to the end and we could probably continue this for another long period of time, but a couple more questions that I had. So let's say that we have an approved therapy, or let's put it differently. Let's put it in a study. So now you and I are figuring out another study because we really want to look. Would you accept, or would you think that this approach would be useful in a patient on an exon skipping therapy? Would you think it would be synergistic with that, or would be additive? What about a gene therapy? Would you include it in a person who had gene therapy and was, X age, or the gene therapy was 4 or 5 years back? So are there in your head people that you would exclude from this therapy should it be approved?
Absolutely not, Pat. And one of the things that we've talked about for a long time is that CAP-1002 is a good player in the sandbox of other therapeutics, but may even actually be the shovel to the bucket, so to speak, because talking about Lee Sweeney, somebody that I respect and admire so much, when I first showed him the data from the first clinical trial, he was enthusiastic because he felt that if the children get a gene therapy or exon skipping and start to have improvement in function, it's going to put more load on their hearts. They're actually going to have to, have a heart that can meet that need. And so any therapy that improves heart function and reduces the amount of scar is sort of a win. Nobody really is talking about that at all. So absolutely. We have done preclinical work with some of the gene therapy constructs just to make sure there's no blocking of the gene therapy effect. And what I can say is that they are specifically not interactive in terms of interrupting a gene therapy effect or a CAP-1002 effect. Both seem.
Okay, that's good to know. I know we have one young man that I think of who fell out of his chair after he wasn't seatbelted in his wheelchair, had a catastrophic accident, was hospitalized for months and months on end, and now he's home, thankfully, and trached, but able to speak and slowly coming back a little. But one of the questions is, he's incredibly vulnerable as an example, and would you think, under those circumstances, that this would be a safe therapy for his family to consider?
Absolutely, and in fact, I think that family might have just reached out to us recently. So absolutely, it's a very safe therapy, and especially since we're doing this work with compassionate use now with COVID, we're very familiar with how to activate that pathway. So I would definitely think that it's worth trying. I'll just make one statement while we're talking about the community, and that is the regulatory pathway is sometimes not reflective of the clinical relevance of data. I understand the regulators want to make sure that every I is dotted and T is crossed before they approve something. They are required to do that by our government. But, this is a situation where community makes a difference, and the advocates know this. So this can be potentially up to you. Please, if you think this is a therapy that you'd like to see approved, join me in getting that done.
Yeah, I think the community has demonstrated that they are actively engaged in the regulatory process, and certainly then once we get things approved, we and other organizations are really working hard to make sure they're accessible. So I'll let you go on and then finish up, and we can round up with it because we have a few more minutes. So instead of making you go back through all the slides, I think we've covered most of the questions and my questions, so why don't you wind up, and then I'll end up with a few last questions, if that's all right with you.
Yeah, I think that's great. And I am actually, you started asking your questions when I was done. I just would like to mostly thank two groups of people. One shown here on the slide is our national PI, Dr. Craig McDonald, who has been an extraordinary leader in this trial, and then also in the development of the plan to measure the impact of the therapy on patients, plus his concern for patients. And every other person listed here are people that are laudables and notables in the space nationally and internationally that have helped move our program forward. So thank you for your advice to me in helping to build this program. And then the PIs at the 9 sites, these are physician leaders who care so much about their patients and the families, and they've worked with us to get it to their patients. Clinical trial work is hard. Sometimes getting these trials started, for those of you that are on the family side, it is literally like watching paint dry. Because there's so much administrative stuff that has to be gotten through, and all of these leaders helped us do that. So thank you to all of them. And, without any more time taken, families, thank you for flying, for sitting in clinics, for patients, for lifting up cups 22 times when you probably don't want to, for breathing into tubes, and all the things that you have to do. What I can say is that every time you do that, you are helping yourselves and your brethren get better over time. So thank you for all of your support.
So Linda, that open label extension, are you going to keep that going until there's an approval? Even if
that's
you have
my
to
goal.
Do a next study? Okay, that sounds great. And you're absolutely right. The heart is a muscle too. We were so, PPMD has been so happy to support Conhor's analysis of cardiac MRI. This is where we're learning about that heart. We're learning what it does over time. We're learning when it gets to that tipping point where the ejection fraction becomes at real risk. And we, much like you, are asking that question. If you preserve ambulation, are you putting the heart at greater risk? So I think that the data certainly is compelling. We all wish you the best, of course, going to the FDA. We're just working with CBER. So there's 2 divisions of the FDA that this community works with: CDER, which is the Center for Drugs, and CBER, which is the Center for Biologics. The Center for Biologics, where we see our gene therapy studies and our cell-based therapies such as this one, and that's the reason for the BLA. So we are very hopeful for your next step with FDA. We're behind you all the way, and we look forward to this therapy and many others in Duchenne. Linda, this has really been good for us and for this community. Thanks for taking time. We wish you Godspeed and certainly success in your next steps with FDA and certainly internationally, because culture, geography should not separate treatments from these kids, and we firmly believe that. So thank you very much.
Thank you so much, Pat. Thank you, community, and we'll look forward to staying in touch.
And thank you all for joining us. This webinar will be posted in the next day or so. and we wish you well. Please stay safe, and we hope to see you soon. Thank you very much for joining us.